Welcome to today's session where we will explore microbiological methods used to assess food safety. In this video, we will focus on aerobic mesophilic plate count, a method which is used to determine the microbial population in food samples. The aerobic mesophilic plate count is important for assessing the general quality and safety of food products. This method helps in estimating the microbial load, which indicates whether a food product is safe for consumption. The materials that we require to carry out aerobic plate count are as follows. We'll require plate count agar, peptone water 0.1%, sterile petri plate, pipettes and test tubes, and an incubator which will be set at 35 degrees Celsius. Let us move to the procedure. First is preparation of food homogenate. So, whatever food sample that we have to prepare a homogenate in such a way that it will be completely miscible in the saline that you will be using to carry out the dilutions. So we'll begin by preparing a 1:10 dilution of the food sample. If the sample is liquid, measure it and mix it with appropriate diluent. For viscous or solid samples, blend or use a stomacher bag that will help in even mixing of the sample. Second step is the dilution process. Prepare decimal dilutions if you are expecting the microbial count to be more than 2.5 into 10 raised to 3 CFU per ml per gram of the sample which has been provided to you. Use a fresh sterile pipette for each dilution to avoid contamination. Pipette 1 ml of the food homogenate in 9 ml of diluent and then continue the serial dilutions as required. Step number 3 is pour plating. We have to label the petri dishes with the number of sample, dilution and the date on which we are carrying out the analysis. After which we will pipette out 1 ml of the homogenate into each plate and then pour 10 to 12 ml of molten plate count agar, and the temperature of this agar should be between 42 to 45 degrees Celsius so that the organisms are not harmed which are present in our food sample. After that, we will mix the contents by swirling and allow the plates to set. Step number 4 is incubation. We have to incubate the plates upside down at 35 degrees Celsius for a period of 48 hours. We have to ensure to maintain proper temperature and time as they are very important for accurate microbial growth. Then we come to counting and results and interpretation. After incubations, we have to count the colonies which are present on the plate. For counting the colonies, we have to take the plates which are having the colony count of 30 to 300. We will record the colony numbers and use the formula which is given below to calculate the colony forming units per ml of the sample which was used. So, as seen in the formula, if the plate contains colonies from two different dilutions, you can calculate the CFU based upon the dilution factor which is denoted by T. The results which are there, they are interpreted as the total number of colony forming unit per gram of the food sample which are used. If no colonies are observed, the result may be expressed as less than 1 into 10 raised to 1 CFU per gram. Limitations of these methods are: one of the main limitation is that microbial cells occur to be found in clumps in the food in which they are growing, which may not be evenly distributed, leading to inaccuracies in the colony counts which we get. Also, the second limitation which is there, that is the APC method which is there, it may not support the growth of all the bacterial species as conditions in the media may not be optimal for every type. So these are the two limitations of using this method to carry out aerobic mesophilic plate count of the organisms which may be found in our food samples. Coming to the conclusion part, the aerobic mesophilic plate count method helps us to evaluate food quality by estimating the microbial content in the food which we consume. It's a simple yet effective tool in ensuring food safety, though it has some limitations that need to be considered. Thanks for watching the video and stay tuned for more insights on microbiological testing methods for food samples. Thank you so much.